screen.
Also. Okay.
Please hide the notifications, sir, please.
Okay.
So you're
Is
good to
it-
go, sir. You're good to go, sir. Yeah.
Yes. Uh, my dear friends, it is my pleasure to be with you today to talk about freshwater fungi. That is my main area of interest. Besides that, we are carrying out several other aspects too with regard to macrofungi and also not only freshwater fungi, some of the fungi exist in mangroves and marine ecosystem. Uh, so this freshwater fungi is a very vast kingdom, fungal kingdom, and everybody is aware that fungal kingdom is parallelly developed along with the plants and animals. The plants have the ability to uh, fix the carbon from photons, whereas animals have lumen to digest the nutritional material in the gastrointestinal tract, whereas fungi, they are not in a position to uh, fix the photons, and also they do not have gastrointestinal tract, but they have a powerful enzyme system and also some of the metabolites. They can produce a lot of metabolites, and they are responsible for degradation of the remaining material. That is, the plant and animal material will be degraded by an array of several uh, fungi. Uh, so the human needs will be normally what people think that it is fungi means it is pathogenic. It is pathogenic human being and it is pathogenic to plants like that. But they do a lot of scavenging work as decomposers, and decomposition of organic matter is one of the prime function of these fungi, and they are very important as food, and food processing can be carried out by that. You know very well about the yeast, how they function, and many of the macrofungi are edible. Nowadays, people are aware that mushrooms are very good nutritional source, and they are producing several pharmaceuticals, and they also produce some of the biofuels and volatile compounds, and they serve as biopesticides because they they occur as endophytes, and then they bring down the pathogenicity by the insects, and they serve as bioprotectants, and also they produce varieties of merchandise that is very useful for industrially useful material like leather, and several types of material can be produced from fungi. I'm specifically discussing about the fungi, uh, especially in aquatic environment. And if you look into the historical perspective, the terrestrial mycology is having history of more than 100 years. But aquatic mycology is very recent, and mostly it is about about 50 to 60 years old, because people have realized that fungi are there in aquatic ecosystem like fresh waters and marine waters and mangrove waters, and they do several important functions in the ecosystem. And uh, the fungal biomass is tenfold higher than bacteria in some of the ecological situations. But you know, basically, bacteria dependent on the surface, but fungi are not depending on the surface because they have mycelia. They penetrate into the material, whatever leaf material, woody material, any other type of material, it can penetrate very easily because of their enzyme system, and they can encash the that is the organic matter that is will be unlocked by the fungi. Therefore, fungi are tenfold higher than bacteria in many occasions, many ecological niches, and it is possible to evaluate the fungal presence in any detritus or any material, maybe food material or grains or any other type of material, by using C14 acetate technique, and that is mainly using the fungal ergosterol, and also thymidine can be used for bacterial DNA. Therefore, fungal production is giving an idea that it is fungal production will be much more than bacteria. And if you look into the annual production of fungal production, will be 16 to 193 gram per square meter. That is, it is going to involve in decomposition and dependent on the availability of organic matter. If organic matter is sufficient, organic matter is available, they are going to transform that and it will involve mainly in the biogeochemical cycles. See, the lifestyle of aquatic fungi, as you know, although they are referred as aquatic fungi, they also occur in terrestrial conditions, maybe semi-aquatic conditions, and sometimes they may produce perfect state, and because they produce a perfect state, they can survive for a long duration in terrestrial situation. Aquatic condition is prominent, and even in anaerobic condition also they exist because they penetrate the mycelia deep in the sediment, and as long as the mycelia is continuous, it can translocate oxygen from the atmosphere or from water and decompose some of the organic material. So that type of feature we cannot find in bacteria, but fungi are well versed with that because the mycelia can run through the sediment, and as long as the mycelia is continuous, it can translocate oxygen from the atmosphere or from water and decompose the material. Not only the aerobic condition, but in anaerobic condition also they can degrade the organic material. And they are halophilic. They can withstand salinity conditions, especially in the mangrove ecosystem and also marine ecosystem. You know very well about mangrove marine ecosystem, where there will be salt content is very high, but some of these fungi have adapted for that condition. And thermal temperature, high temperature tolerating fungi are there, and they are barophilic. As I mentioned, barophilic means it is the pressure loving fungi. So the deep sea has a lot of pressure and also low temperature, and they can withstand the low temperature as well as the pressure, high pressure, and they perform several functions. And they also associated with the plants and animals. You know, plants, they are associated as ectomycorrhizae and endomycorrhizae and endophytic fungi, and several type of associations you can see not only on the surface, but it can enter into the cell proper. Some of them enters into the intercellular spaces. That is, the ectomycorrhizae net will be formed, and arbuscules and vesicles will be formed by arbuscular mycorrhizae, like that. With animals, they see in our intestine, the highest number of organisms exist in intestine is bacteria. The second group is protozoans, and third group is fungi. Some of the fungi also exist in animal intestine. They perform certain functions, especially yeast and such type of things. And they also associated with micro and macro algae. They are symbiotically associated. For example, seaweeds are associated with different fungi, and they are also mycoparasitic. So the fungus is going to colonize another fungus and try to transform some of the nutrients, like that. That is my reference to mycoparasitic. And freshwater fungi, if you look into different divisions, the Ascomycota that is one of the groups, and Chytridomycota is another group. You know very well, chytrids are detrimental to anurans. A lot of hue and cry was there about how chytrids are responsible for deterioration of frogs, like that. But it is maybe partially true, but chytrids are very, very important. Even in the aquatic situation, they are important. And some of the lower fungi, especially Saprolignia, they are all affecting the fishes, and fish population will be affected by Saprolignia and such type of lower fungi. But people are interested to evaluate such type of pathogenic fungi associated with the fishes and such things, rather than other organisms. They are present in the aquatic ecosystem and functioning in a different way. Like, it is not exactly the only this. I have already mentioned that all fungi are not pathogens. So a lot of useful fungi are there in and around us. And Cryptomycota is another group, and Zygomycota is another group. These are the 4 divisions we can find in freshwater. And the lifestyle, what they perform, what they contribute to the nature is their saprobic. The dead and decayed material will be transformed. So the biogeochemical cycle will be operated by these organisms, and then they are planktonic in fresh water because they are floating. They are floating in water and they catch the organic material very easily because of their different types of fungal, that is the spores or the conidia produced by them. And many of them are endophytic with the plants, riparian plants, that is the trees, the plants present in the marshy region and also in the riparian region of the stream, because many of these trees and plants, they produce roots into the water, and those that is the stagnant refuge for several freshwater fungi, and some of them are pathogenic too. It is not totally, they are not major pathogenic fungi, but some of them are pathogenic under aquatic condition, and some of them are parasitic. How much is global freshwater? Because if you compare saline water and freshwater, the saline water is 97%, and freshwater is approximately 2.5 to 2.7. We can say approximately 3%. But this 3%, nearly 1.75 to 2% are locked in glaciers and ice and snow. And another 0.5 to 0.75 is locked up in the groundwater and present in soil as moisture. And we are getting only 0.01%, that is surface water in lakes, rivers, and swamps. Because this is very, very important for us, because we cannot live without fresh water. So therefore, it is high time that we should pay attention towards the importance of fresh water and its usefulness, and also type of fungi they exist here and what are their roles in freshwater ecosystem. The freshwater fungi are also referred as Ingoldian fungi because Ingold was the person in 1942, out of curiosity, he had a small creek behind his house, and he took some foam sample, whatever foam that is accumulated in water, he collected and observed under microscope. For his surprise, he could find several conidia, different shapes of conidia. Of course, in the late 19th century, people have classified some of the fungi based on the morphology of this into algal members. Afterwards, they realized that they are not algae, they are fungi. And the correct shape was given by Ingold in 1942. And then he has given that in honor of him, it is referred as Ingoldian fungi. So in the freshwater, they transform plant litter, and also energy flow takes place. You know very well about how energy flow takes place. So the importance of detritus food chain detritus food chain has been less studied compared to that of primary productivity and secondary productivity. We are all well versed with the photosynthesis and production like that. And then the secondary productivity by various type of organisms. But the detritus food chain has been least studied, and these organisms are the one they are involved in detritus food chain and transfer energy to higher trophic level. So I will come to that point sometime later. And the number of species are highest in the mid altitudes of the Western Ghats, and even probably in the Himalayas also. It may be true that mid altitudes have good repository of freshwater fungi, especially aquatic hyphomycetes. I am dealing only with the aquatic hyphomycetes, not with other organisms. And the population of these organisms will be similar in different climatic zones, and therefore it is possible to guess the latitude and longitude based on the occurrence of these organisms in a particular stream location. So that's what Ingold mentioned, that if you take a foam sample from a location and analyze, come to some conclusion that we can approximately guess that this is the latitude and this is the latitude of the particular location, like that. So that is the occupation of these organisms are very important with regard to environmental monitoring is concerned. And so far, total morphospecies now from 335, the number has risen up to 360 based on this is the statistics of 2016, and now it is after 8 years, now it is almost close to 360 number of species. So this why I am mentioning that it is neglected mycota, because not many people in India are working on this group of fungi. So there are only 2 or 3 groups working. So one of the groups is from our laboratory from Mangalore University. From way back 1978 onwards, we are concentrating on this group of fungi. And another group is from Nainital. That is from Nainital, Professor Sati and his group are concentrating on this group of fungi. And these are the major region, that is the Western Himalayas, and also the mid-Western Ghats are the locations where more work has been carried out on this group. And the rest of them are, we can get some sporadic information from middle, that is Madhya Pradesh and such regions, and even from Tamil Nadu and Kerala, and even northeastern region. They are all very sporadic, not very extensive studies, except Nainital and also from Western Ghats. These are the, as I mentioned, they are the key fungi and in the aquatic ecosystem to decompose organic material. And they also occur outside the streams, not only streams, they also occur outside the streams. And the diverse substrates will be tackled by this group of fungi. For example, not only leaf litter is very fragile, and they also colonize on the woody material, because that wood material is going to stay a long time in water. Therefore, it can entangle somewhere in the crevices, and it is available for that perpetually in providing nutrients to them. And also these roots of different plants also stagnant refuge for them. So therefore, they are colonizing various, and even they can occur in the sediments and other type of detritus available in that. And also some of the macrophytes, some of the aquatic plants also colonized by these organisms. And in fact, this group has been considered as a model fungi because you are not dealing with single organism. You are going to deal with a community, fungal community. So if you study about one fungus, you cannot come to conclusion about the contribution of this fungus in the ecosystem. Instead, it is a community. You may get some 40 or 50 species or 100 species. So if you go to Western Ghats and collect these organisms, in one collection, you will get up to 80 to 100 species, you are going to get it. That is one of the hotspots of this fungi, this group of fungi. And you are going to deal with the community rather than single organism. What happens to this community if the land use pattern is changed? What happens to these organisms if pesticides are added to the agricultural fields? And these are all some questions to be answered. And part of that has been tested. And water quality assessment, these organisms are important because they are going to, so they do not allow any organic material to stay for a long time. They are going to decompose and they are going to recycle it. And pollution assessment can be done with this. And as I mentioned, Western Ghats is one of the hotspots of diversity, and even Central and Western Himalayas also have a lot of fungi. And from India, so far about 20 species have been reported. From our group, we have reported 5 to 6 species, whereas the Sati et al., they reported several species from Nainital. Several new species have been reported. So this you can see the green and red and yellow and also partially yellow dots here and there. Fortunately, we have several dots here in Western Ghats and mid part of India, and also the Himalayan zones. They have indicated. So some work has been carried out from our India, and also you can see a lot of the observation here that the European region is giving high priority for the aquatic ecosystem and its health. Therefore, a lot of work has been carried out on this group of fungi, and even North America, Canada. These are recently You can see some of these studies in the Brazil and South American region also it is there, and some African region and Australia, a little bit of work, and even Southeast Asian countries also concentrating on this group of fungi because they are very important in freshwater ecosystem. So as I mentioned, they do not produce spherical type of course, they produce a few fungi produce spherical or conventional shapes like elongated or fusiform type of spores, but many of them produce uh, varieties like a sigmoid or half moon shape or little moon shape. Like these are all some of the sigmoid shape, and also triradiate and tetraradiate type of spores. And so it is not, see, this is a fungal spore, and it is highly branched, primary branch, secondary branch, and tertiary branch, like that. It is sometimes it is not possible to believe that it is a fungal spore. So this is a single fungal spore, and some of them looks like shrimps. Like see this spore. This is the point of attachment, and it has 4 arms, and it is encircled. That is Garofilla. And sometimes it is not possible to believe that this is a fungal spore. In fact, it is a fungal spore. So this is another one which central portion is thick, and the arms are spread like that. And these are all different patterns of fungi, conidial fungi we can find in the aquatic ecosystem. So 3 types of spores will be produced, conidia will be produced by these groups. That is colecoid spores. Colecoid means it is the sigmoid type of spores. See, S-shaped. And the S-shaped spore, if it fall on the surface of leaf or wood, 2 points are going to touch. So that is, you imagine the 3-dimensional view. This is the tip, and this is the base. And this is going to touch the bottom. This is also going to touch the bottom. By that, these are the regions where fungus produce aprosoria and colonize the leaf material or woody material, any type of organic material. You can see some diversity. See, this is a sort of Z-shaped fungal spore. These are all some hockey stick-like spores, like that, variety of spores under colecoid spores. Second group is stauroid. Stauroid refers to the multiple, that is the multiradiate, triradiate, tetraradiate, multiradiate, like that. And some of these spores in the Western Ghats, I have seen one fungus has 22 arms. It is something like a very interesting spore. And see here the variations with regard to septation, and it is this is a sort of adaptation to the aquatic environment, just like that of colecoid. And this is the tetraradiate, triradiate, and multiradiate, because they are leading a planktonic life in the aquatic system, because it can float because of these appendages. It can float and also catch the organic material very easily. So it is something like, if it drop on a surface, it is something like a tripod, like 3 points are going to touch the substrate, and that region is going to germinate and encase the organic material. The third group is Helicosporus. So Helicosporus, they are twist-like thing. They are encircled. Here I have some other. This is a triradiate, tetraradiate, triradiate. You leave this, and others are the Helicosporus, and this is an adaptation where they can trap the air bubble. Because they trap the air bubble, they can float in water, and they carry from one region to another region. They are referred as Helicosporus group. So if you look into the capability of these organisms to produce conidia, because they are known to produce enormous number of conidia. Any fungus you take, they produce lots and lots of spores will be produced, and it will be disseminated. So this one of the reports in 1964 by Nielsen says that 300,000 conidia per leaf, and our average leaf is going to produce 300,000 conidia. So it is not a small number. Similarly, in 1981, Webster Descos reported that 30,000 species, 30,000 conidia present in 1 liter of water. So that is another amazing thing, that what do they do? So many spores are present, so many conidia are present in water. What are their role? That is how the curiosity has been started. And the further 1992, Berlocher and his group has mentioned that that is per leaf it can produce 1 million conidia. That is equivalent to 8 spores, 8 conidia per microgram of leaf, like that. So this the beauty of this work is conidial enumeration is very important. If you enumerate the conidia, how much of conidia is there in water? How much of conidia produced per gram dry mass of leaf material? How much of conidia produced per gram of live root? So this is the sort of enumeration, and it is equivalent to that of calculation of ATP. How much of ATP is there? It is equivalent. It is not. If you enumerate the quantity, that is enough. That is equivalent to that of ATP. If you do ATP work and then if you say this much of ATP is there, it is equivalent to that of quantity enumeration. Another method is you can estimate ergosterol. Ergosterol is a type of lipid. You know very well that human beings have we have cholesterol and plants have stigmosterol, whereas filamentous fungi have ergosterol. Ergosterol is a signature sterol, and if you calculate by HPLC, ergosterol 5.5 milligram is equivalent to 1 gram of fungal biomass. So this is the conversion factor. Even if you calculate ergosterol and then say how much of fungal biomass is present in a detritus or whatever type of material, you can get it from the aquatic ecosystem. So what are all the niches they exist? So they submerged plant detritus is a very common substrate, and flowing water, they are planktonic because they float, and hyporheos, that is sediments and biofilms, because algae in water, they produce a sort of film on the surface of rocks and other stagnant surfaces. And the hyporheos, where aquatic fungi can colonize because that is sticky and they can grow there. Like, and you can also expect from foam and scum, they are going to accumulate. That's how Ingold has collected these organisms, because in the foam, wherever their water is swirling, a foam will be developed. This foam consists of a lot of conidia, and that can be identified. And they also, the coarse particulate matter and fine particulate matter, also some of the parts where you can expect in the sediment also, you can expect this fungal association. And also riparian roots, as I already mentioned, hydrophytes, and stream borders, a little bit partially aquatic, and intermittent region, temporary streams, intermittent, and stream slopes are another area where a lot of organisms exist there, either in the perfect state or imperfect state. But when it comes to stream, it will be going to become imperfect and try to lead the life like that. And if it is transferred from stream to outside, there are some chances that they may produce perfect state and it will stay for a long duration. Animal intestine, if you take fishes and invertebrates, you can, in the intestine, you can see some of the conidia. They are live conidia, and that's how they will be transmitted from one place to another place. And they also exist in estuaries, mangroves, and delta region like that. And even in the tree canopy, also you can expect a lot of this group of fungi exist there. But I need not necessarily explain too much of this, because this is the leaf material that is allochthonous leaf material or autochthonous leaf material. So this is the mycoflux, where the fungi are going to colonize, and they are going to convert into fungal biomass. And this again, it is in the lower portion where it is referred as benthic shunt, where the invertebrates, macroinvertebrates will be benefited, and then they are eaten by some of the plankton, zooplankton, and lot of phytoplanktons are there. Phytoplankton, like that, even aquatic freshwater conidia also eaten by the zooplankton, and then these benthos are important for fishes. That's how you can expect the sediments consists of several fungi, and also the water column consists of several fungi. They are interacting with other organisms like microfungi, zooplankton, and parasitic fungi, and even macroinvertebrates, and seaweeds, and such type of things, or macrophytes in fresh water. So what are the ecological services of this group of fungi? So the major service is decomposition. So you know very well, see how we are worried, especially in the urban region, how to dispose of the whatever this whatever garbage what we get it. So it is very difficult in urban area. But in rural area, it's okay. We can put into some of the trees and plants like that. It will be decomposable. If it is not decomposable, then if it is plastic, then we have to take extreme care about not to introduce into the system to the ecosystem, because we will be suffering because of these microplastics and such type of things, and it takes a lot of time to degrade. That is decomposition, the major contribution like that. And in the detritus, there is an estimation that 63 to 100% fungal association will be there. That means definitely more than 50% will be decomposed, that is transformed by the fungi. And as I already mentioned, that fungal production is much more than bacterial production, because bacteria is surface oriented, whereas fungus is not surface oriented. It is penetrating the detritus material. And fungal biomass, if you look into fungal biomass, 18% of the total detritus mass consists of fungi. 18%. So that is a very big number. And up to 80% of fungal biomass allocated for spore production. So if you take a leaf material, cut into small pieces or cut into small discs, and clean it and put it in distilled water and aerate it with aquarium aerator, then what happens after 48 hours, you can get big crop of these spores. Varieties of spores can be produced like that. So therefore, 80% of these will be converted into conidia. Therefore, our estimation will be on par with the ATP estimation or ergosterol estimation. And it is very important for the food web, because they are palatable for the aquatic insects. And many insects are depending on this group of organisms, because this ergosterol is very, very important, because ergosterol is present in this in the detritus. And many of the insects, what they do, they lay the eggs in the aquatic system, and these eggs are going to hatch and become young ones, juveniles, and they start eating the detritus material where fungus is there, because this ergosterol is very important for its metamorphosis. And after its maturity, it is going to become arboreal. It is going to go away from the stream, and again the adults, after mating, they are going to lay the eggs in the aquatic ecosystem. Like that, the metamorphosis of several insects dependent on the ergosterol of this group of fungi. And also they are very important in energy flow, because whatever freshwater fishes we are getting and freshwater products what we are getting is mainly the contribution from this group of fungi. And this slide shows you the vertical diversity. As I mentioned, vertical diversity, leaf litter and its decomposition. And these are all microbial decomposed. This is the detritus material. This is the microbial loop where decomposers are there. And these decomposers will support the detritivorous invertebrates and such things, and then predators like. And the vertical diversity, whereas horizontal diversity converts leaf material into the dissolved organic matter like that. And there will be some competition for this detritus with invertebrates as well as fungi. And therefore, this material will be, some of the invertebrates wait till the fungus colonize, because they will be more benefited if the fungus colonizes. Otherwise, raw food, raw leaf material may not be palatable for them. Therefore, they wait till fungus is going to colonize. By that, they will get several bioactive compounds from that. As I mentioned, decomposition is everyman's response to what fungi do. That is this is the question raised by Christensen in 1989. Because we are neglected this area. That's why we are suffering that how to dispose our waste material in our urban area, even rural area. Sometimes it's difficult. Like they produce a lot of structural polymers, cellulose, hemicellulose, pectin, lignin, and such type of these polymers will be degraded by this fungi. And changing the land use. If you change the land use, for example, perturbation, especially with regard to addition of pollutants and channeling. So if you make a stream more uniform, like a channel, it is not much diversity you cannot find, but it should have a lot of boulders, a lot of discrepancies there. By that, what happens? The leaf material and wood material will stay there, and it is necessary fungal is going to colonize, and then it is going to continue the eutrophication, and all these things will happen, and the climate change is also have a lot of impact on this group of fungi, and this is referred as mycoplankton because they are going to float in the aquatic ecosystem. So here is the major pathways of fatty acids in food chain. That is the linoleic acid will be converted by higher animals, converted into arachidonic acid. So this linoleic acid will also be taken up by algae, fungi, and plants. Especially even fungi also do this. That is alpha linolenic acid, and alpha linolenic acid to eicosapentaenoic acid, and eicosapentaenoic acid to docosahexaenoic acid. You all know what is the importance of docosahexaenoic acid. So the docosahexaenoic is very important for brain development in children, and that's how the transformation of these fatty acids will be taken place by this group of fungi in aquatic environment. And in the studies in the Western Ghats, we have reported nearly 100 species belong to 55 genera, and that accounts to 30% of the global report so far. Richness increases high altitude and mid altitude. So richness is high in the mid altitude rather than high altitude and low altitude. That's what, and even lignicolous fungi, that is especially ascomycetes, we have reported nearly 60 species. And aeroaquatic fungi, not much work has been carried out on aeroaquatic fungi. This is one of the areas where people can concentrate. So mid-altitude streams, it is 500 meters above sea level, is ideal for several organisms. That's what I mentioned, that one trip, it is possible to collect nearly 80 to 100 species from this area. And also we can screen water, foam, and leaf litter like that. Water can be filtered through the millipore filter and then stain with aniline blue in lactophenol, and what are the spores present in that can be evaluated. Foam can be fixed, and it can be brought to the laboratory and study that. Leaves, as I mentioned, it can be studied how many species are colonized like that. So we can find these organisms in the canopy, and the post-monsoon season is the best season for these studies. And several conditions, you can think of temperature, pH, conductivity, water quality can be tested, and dead substrates like leaf, bark, wood, root litter, these are all recycled in the aquatic ecosystem. And live parts of hydrophytes, roots can be studied. Lot of methods are available. Water filtration can be done, direct observation can be done, damp chamber incubation can be done, bubble chamber incubation can be done, and latex trapping. We developed a new technique to trap this conidia in the aquatic system with flowing water. What you should do is you take latex of banyan or latex of artocarpus, jackfruit, that can be smeared like blood stain, blood smear like that. You can smear it, dry it, and put into the slide box and make some openings and hang or put it in the stream. Whatever the spores that is releasing, some of them come and they settle on the latex, and this can be retrieved at different period, maybe once in 3 hours, once in 6 hours, or once in a day, like that. It is possible to use this latex trapping technique. Is one of the authentic techniques, can be used for observation. It is a very simple method. No cost is involved. If you have some slide boxes and latex, it is available everywhere like that. So this is one of the hotspots of Western Ghats is Sampaje stream. So 90 species, I have mentioned already, 80 to 100 species. It is 27% of the total reports what we get in global report. And see, latex trapping 24 hours. In 24 hours, we can get nearly 50 species of conidia. Like that, it is not a very simple and very elegant technique to study this group of fungi. Another study in the Western Himalayas, Himachal Pradesh, that is especially Nainital, Suresh and Suresh Sati et al. They have studied lot of aquatic hydromycetes as root endophytes, more than 30 species. In addition to diversity, taxonomy, role of aquatic hydromycetes, they studied the root endophytes. So these root endophytes have some role. And what are the importance of root endophytes? They have studied some of the specific species as colonized into some of the plants, and they are important in plant protection. And plant growth has been promoted by these endophytic fungi. And some of these fungi are very good phosphate solubilizers in aquatic system and provide to the plants. Like that, as you know, phosphate is not easily available, and it will be in the rock phosphate, and it has to be transformed into orthophosphate, then only plants can utilize. And therefore, phosphate solubilization is one of the important functions of endophytic fungi. And also, they have reported several bioactive metabolites important for industrial purposes or bioactive metabolites used for various purposes. So pollution is causing a lot of damage because of global warming is one thing, and pH is going to change. But usually, this group of fungi grow in little bit of acidic pH. Somewhere between 6 to 7 is all right for them, not below or not above, like that. Although some of them are withstand the pH. So 6.5 to 7 is very good. And conductivity and domestic and urban pollution, water is included, and agricultural chemicals are leached into stream ultimately, and several industrial activities are, and also some of these exotic tree species are growing. Eucalyptus, acacia, hevea, silver oak, these are all rubber, and these are all exotic tree species, but still fungi are going to take up and utilize it as an organic material, but the riparian vegetation is totally changed by this group of exotic species may not be good. We should have some native tree species will be always good. Destruction of riparian vegetation is another problem. Nanoparticles, you know very well, very small nanoparticles are affecting this, and xenobiotics, a lot of pollutants are affecting these organisms. And so this biodiversity in ecosystems can be, that is structure and function. So how this community structure exists in the aquatic ecosystem, and what are the functions they play in the aquatic ecosystem. That is litter breakdown. It is a simple, authentic, and cost-effective method. Monitoring, that is guidelines to control human impact. So if it is human impacted, highly polluted region, whether they exist, but the number will be less. Diversity will be very low. Very few species are resistant to pollutants. Like, so what are they can be used as indicators of pollution, and also pollutant level can be indirectly, one can go for pollutant sensitive and pollutant resistant organisms can be found out, because we are destroying the freshwaters like anything. Therefore, this group of fungi will be very helpful to recovery of this whatever the stream that has been already spoiled. It can be recovery can be tested using these organisms. And also you can go for that is if you go for pyrosequencing P454, see the whatever species you are going to report. If you say it is 100 species, if you go for pyrosequencing, it is doubled. It is going to become 200 species. Therefore, what we know is little bit, but molecular studies will be very useful. Like, so there are several approaches. That is population. We are dealing with the population, not with single organism, a community. This community is going to change over a period of time because of human interference or climatic change or the other type of perturbations. Therefore, the community ecology, community study will be more valuable rather than studying a single species. Like, and also decomposition. Decomposition, they are going to stable, they become fragile, and they become dissolved organic matter. Like that. Quantitative studies can be applied. Induction of perfect state can also be performed by that, because some of the wood material will be colonized in the aquatic ecosystem. Those wood can be taken out and that can be dried in the laboratory, But this drying, instantaneous drying may not be useful, but instead slow drying process. Instead of sudden drying, if you just dry it step by step, then there may be possibility that they produce perfect state. So that can be seen in some of the root colonized endophytes, and also wood colonized fungi can produce perfect state. And prove only 30 species have been reported so far. The connected imperfect and imperfect connections has been studied. And canopy is another very important niche of these organisms, like tree holes, through fall and stem flow. So during rainy season, there will be through fall. Through the canopy, water drips out, and stem flow can be collected and studied. So these are all some interesting aspects. And aeroaquatic habitats, where there is a little bit of moisture is there. But so many fungi, they are in the aquatic situation. When the substrate is exposed to air, they produce conidia, like that. They are referred as aeroaquatic. Root endophytes, stress tolerance, and they are useful for the bioremediation purposes. Mode of transmission, how do they get transmitted? Is birds are transmitting, invertebrates are transmitting, or human beings are transmitting, or what is the way it is transmitted from one region to another region, like that. So that has to be a very interesting one. So there are several simple methods can be done. So one need not necessarily to use a very sophisticated instrumentation. Such things are not necessary. Simply you take water and filter it, and see whether there is any conidia. Especially you need a millipore filter of 5 micron or 8 micron filter. Use a syringe of 25 ml. You draw the water, 25 ml water, and pass through this millipore filter, and then you stain with aniline blue in cotton blue in lactophenol, or aniline blue in lactophenol, and keep the slide in dark. And whenever you find time, maybe after a week or so, you can scan whether there is any conidia, because the conidia are very bizarre. It is easy for identification. Another method is foam. You can collect it. Anybody can collect foam and fix it in formalin acetic alcohol, and then bring to laboratory and study. And conidia, you can count the conidia, maybe 100 or 200 or 500 conidia, and you classify how many species are there, like that. It's a simple method. If you have a simple microscope, in high school also one can do this. Not necessary that we should have a very sophisticated laboratory. Leaf incubation, bubble chamber incubation, I will show the photo later. And the leaf baiting can be used. Leaf can be introduced into, sterile leaf can be introduced into the stream, and see whether they are going to colonize, like that. And as I mentioned, the latex or the rosin. Rosin is from the gymnosperm, some polysaccharide-like material. It is coated sites. That has been studied. Instead of rosin, what we did was we used the latex. So this latex works very well, because we did some diurnal study in the Western Ghats and coastal region. Every 3 hours, you are going to retrieve the slides and see how this population dynamics is going to change, like that. So therefore, it is not a very expensive technique to follow to study this community ecology. And there are, that as I mentioned, conidia by filtering, mycelial biomass can be estimated. Epifluorescence microscopy can also be followed. And as I mentioned, latex trapping can be followed. Then indirect methods are there. Particle plating can be followed. Foam induction. So you take the leaf material, incubate it in water for about a few hours, and then you add a small quantity of detergent and aerate it. By that, what happens? Foam is going to accumulate. So this foam is going to ultimately collect the spores, and then one can study what are those spores like. Ergosterol can be estimated using HPLC. Chitin can be estimated, another method, because the cell wall of fungi consists of chitin, and how much of chitin is there in the detritus will tell you about the population. Mass loss can be studied. How much of mass of leaf material will be lost over a period of time. That is, what is the half-life of leaf will be transformed by this particular environment, and also like that. And the transplant experiment can be done. Very interesting, because transplant experiment means, let us assume you have a polluted stream. Another one is fairly not polluted, may not be totally pristine, but you can allow that to colonize for about 2 weeks in both the one, and afterwards you switch this from pristine to polluted to fresh water, like that. So it is possible to study the dynamics of these organisms and how it is going to change over depending on the conditions, like. And molecular approaches, as everybody is aware nowadays, DNA barcoding can be done, pyrosequencing can be done, ecosystem service. There are a lot of ecosystem services by this group. That is, we have to link the structure and function, and next generation sequencing NGS can be followed, and blend of conventional and molecular studies. So we cannot leave the conventional studies, and molecular studies is going to give you more insight, and both blend of these studies will be more beneficial to study this group fungi. So as I mentioned, it is possible to study model mycota in mesocosm and microcosms. Mesocosms and microcosms, something like you are going to mesocosm refers to the stream, like, and microcosm in the laboratory level, laboratory where you can have an aquarium where you can put some of the leaves and inoculate fungi, and then allow them to grow, and then you can introduce some of the fishes and invertebrates, and whether they feed on that or not, using different types of microorganisms in the field and laboratory, that can be done. And this organism, you can see, as I mentioned, it is going to produce different types of conidia. That impaction, that is attachment to the surface, is one of the features where a procedure will produce. It can be taken as a variable. Colonization and growth of the organism on a substrate. Thirdly, when it colonizes, after some time it starts sporulating. When the conditions are going to become little bit adverse, like little bit of temperature, like that, and spore dispersal can be taken into consideration, and also spore germination also can serve as variables to study this group of fungi. So the several conservation strategies have been reported. I'm going to complete in another 5 minutes. Cryptic species, they are very rare species. Core group fungi, they are more than 10% of frequency of occurrence. Keystone species supporting other organisms. Along with that, quantitative studies, in situ conservation, ex situ conservation, and also many repositories to have this repository of these fungi. Identification of hotspots of fungi. This is very important because we are losing our ecosystem. So it is very good to study some of the national parks where the pollution level is less and the human interference will be less. And the new and novel strategies can be developed. Habitat loss, and what is the impact of habitat loss, like that. And as I mentioned, keystone loss of keystone species also affecting the aquatic fungi, because if banyan tree, banyan is a very good material, or even rubber leaves is very good material for colonization of fungi. So the banyan tree is a keystone species. If you lose that keystone species, there are a lot of different species are there under banyan, and if you lose it, means it may be detrimental to ecosystem, because keystone species are going to support varieties of organisms like termites, insects, endophytes, and bats, birds. You name anything, it is the keystone species are going to support. And any outliers. So like some of them are maybe the Sampaje in the Western Ghats is one of the site where it is very important, like that. Even Himalayas, is it possible to find out a location, or in Maharashtra, or in Madhya Pradesh, or in south, that is northeastern part. So any location can be, a type locality can be determined, and then that can be taken care of with regard to this group of organisms, and also rehabilitation tasks. There are several gaps. Wood and sediment study, overlapping habitats are there. Most productive outliers, mid-altitude, as I mentioned, several unidentified conidia. So if you study, let us assume that you are going to get 40 species, but out of 40, 4 or 5 cannot be identified up to species level. That means 10% is not identifiable, means they are aquatic fungi, but they are not identifiable. That means they are new species, like that. There is a lot of cryptic species. They are very rare and hidden somewhere. Symbionts, as I mentioned, similar to endophytic fungi, core group fungi, more than 10%. And keystones, among the fungi, there will be some keystone species. They are supporting other organisms in the group. Perfect imperfect connections can be established. So there are two important unsettled questions, that is pattern of diversity, alpha, beta, and gamma diversity. That is regional, community level, cosmopolitanism and endemism and evolution of these organisms are not well studied. Integrated approach, that is blend of taxonomic, phylogenetic and functional diversity in global scale with metagenomic approaches these are the two unsettled questions. So very recently we come to know that there are some freshwater lichens. It is a new ecological niche. On the boulders, on the rocks, you can find varieties of these are the pictures or the lichens, and they are aquatic lichens, and then freshwater ascomycetes are there here, and a lot of fungi may colonize, aquatic hyphae may colonize because it is going to be recycled. So this will be colonized like that. So there is another ecological niche in the freshwater system yet to be studied, because this may be useful for others to study that. There are several tasks ahead. I have already mentioned many of them: rehabilitation and I think riparian vegetation, and also the stream catchments, agriculture, plantations, and pollution, and such type of things. I am very thankful to Mangalore University. It has sustained me even after 10 years of my retirement. Still, Mangalore University wants me to be in the Department of Biosciences, and they are supporting me, whatever little work I am carrying out. I'm thankful to Sri Venkateswara University for this opportunity to speak to the audience. If you have any questions, you please send it to my email ID, kandikere.manasa@gmail.com. Whatever possible, I will reply you. And the organizers of this seminar, especially Bellai and the university authorities, they have conducted this seminar. And also MycoAsia is our own association where MycoAsia and MycoIndia, they are very important. That is very recently they have been established, and they are supporting and also they are inviting a lot of people to give the lectures and educate the youngsters with regard to various types of aspects pertaining to mycology. Thank you very much for your patient listening.
Thank you, sir, for your valuable talk on freshwater fungi.